Hey everyone, we have spent a lot of time looking at letters on screen, H, T, C, and G's. But if you're looking at a genome with millions of bases, you cannot just read it. You need to see it. So today we are going to create our first genomic visualization. We will take a DNA sequence and plot its GC content using a technique called a sliding window. By the end of this video, you will be able to turn a raw FASTA file into a professional looking graph using Python's Matplotlib library. So let's get visual. So the concept is if we calculate the GC content of an entire chromosome, we just get one number, like say 42%. But that's kind of boring. In reality, some parts of the genome are very GC rich and other are GC poor. So these variations often point to where the actual genes are located. To see this, we use a sliding window. We look at a small window of say 100 bases, calculate the GC content, move the window forward by one step and repeat it. And this creates a trace of the genome's composition. So let's begin. First of all, we will have to install Matplotlib, which is the gold standard for plotting in Python. So, and we will also import BioPython. So first let's try to install Matplotlib. Okay, so Matplotlib has installed. Now we will import it and then we will get started. Import matplotlib.pyplot as plt. Then we will import BioPython and one more bio.sequence we will import GC fraction. Right. Okay, so we will load a sample sequence here. If you don't have a large FASTA file, you can generate a long random one or use the random one from our file that we are going to use here. Okay, so now let's write the logic. Uh, we need to slide across the sequence and store the GC percentage for every window. So first we will load our sequence. Record is equal to sequence io dot read my sequence dot FASTA and the format is FASTA. Right, and then sequence is equal to record dot sequence. Okay, we will assign the window size is equal to 100. GC values we will leave blank for now. Okay, now the sliding logic for I in range length of sequence minus window size plus 1. Then inside we will do subsequence is equal to sequence I plus window size. Okay, that should do it. Now we will calculate GC fraction and convert it to percentage. So GC val is equal to GC fraction subsequence into 100. GC values dot append GC val. Right, that should do it. And then we will print calculated GC values, um, GC data points, right? They should do it. So let's see what happens. Okay, so what happened here? So basically, we took a slice of the sequence from index I to I plus window size and then calculated its GC content using BioPython's built-in GC fraction that we imported here and appended it to a list. This one. Now we have a list of numbers ready for plotting. 35 GC data points to be exact. Right. So now for the magic, let's use the Matplotlib library to turn those numbers into a graph. So how to do that? plt dot figure figure size is equal to let's go with something like 12.5. Then plot GC values. Then we will assign it a color. Let's say royal blue. And yeah, we need line width also. If I am right, line width is equal to 1. Then we will add labels and styling. Title, uh, GC content distribution, window size is equal to window size and base pairs, right? Then we need labels for the axis. plt dot x label. It will be position in genome. Yeah, position in genome in base pairs, and for the y-axis, I think, yeah, y-axis, plt dot y label GC content in percentage. Okay, and yeah, we also need a grid. plt dot grid, assign it as true. Line style, we can go with dashes like this. What else? Yeah, alpha is equal to 0.6. And finally, plt dot show to plot the graph. Okay, let's see whether it runs properly. And there we have it. Instead of a wall of text, we see a landscape. Those peaks you see, those might be areas of interest, like CPG islands or highly stable regions of the DNA. This is exactly how researchers begin to map new genomes. So if the graph looks too noisy, you can simply increase the window size. A larger window smooths out the line, shows broader trends, while a smaller window shows every tiny detail. Since our sequence file is very small, so we don't have any issues like that right now. But if you take a bigger file, you can do that. What I suggested. Okay, so data visualization is a superpower in bioinformatics. Today, we learned how to implement a sliding window algorithm and plot genomic data using Matplotlib. You are no longer just a coder, now you are a data scientist, basically. So in the next video, we are going to take things up a notch. We will see how to find motif specific patterns in DNA, like the TATA box, and tell the cell where to start reading a gene. The code for this plotting script will be available on my GitHub, and if you found this helpful, give it a like and subscribe for more, and tell me in the comments what are the genomic features would you like to visualize. See you in the next video.